Today, my assigned topic for discussion in the breast cancer session was an assignment that was really a challenge given to me by the program chairs, Dr. Reyes and Dr. Santos. They gave me the title of Update on Translational Research in Breast Cancer, which is a very broad topic, but it gave me the freedom to discuss and consider some possibilities that might not cross a routine clinician's mind in a busy day. And so I focused in the area of diagnostics and challenges that we face now in diagnosis and treatment of breast cancer. And to this end, I discussed a paper that just was published by one of my colleagues at Stanford, Dr. Christina Curtis, who looked at the evolutionary dynamics of outgrowth of different subpopulations of mutant tumor cells in solid tumors, breast cancer included. And basically, she explored how these dynamics come into play under various selection pressures, like selection pressure from our therapeutics. And basically, with this model framework, you can superimpose actual data from solid tumors and sort of deduce what the types of selection pressure might be, whether it be neutral or positive or negative or some combination thereof. And this is very important because tumors are collections of highly heterogeneous groups of tumor cells with different types of mutations within the tumor itself. So it's important probably to do multiple samples of a tumor because as these cells outgrow, there's spatial heterogeneity as well that evolves. The other topic I discussed was Dr. Haruka Itakura's paper in Science Translational Medicine, looking at integration of digital image data sets along with molecular expression array data sets. So it's now possible through bioinformatics tools to integrate digital image data along with, let's say, gene expression array data and have more interesting and complex phenotypes of tumors that tend to cluster together and have particular suites of genes that are altered within each cluster. And that gives us the possibility of new diagnostic modalities and perhaps new therapeutic treatments that has a more comprehensive tool to integrate all of the available clinical data. I moved from there to a consideration of how all of the multi-gene assays have evolved over the past 5 to 10 years. And most of them started with gene expression array, that is looking at all the messenger RNAs that are upregulated or downregulated in a given tumor sample, and those patterns inform the intrinsic phenotypes of breast cancer, which has therapeutic implications. The steroid receptor positive phenotypes are treated with hormone therapy, the HER2 enriched phenotypes are treated with HER2 targeting agents, etc. But what was interesting in comparing all of the available commercial diagnostic assays that are multi-gene tests in human breast cancer, a paper was recently published by a British group called the OPTIMA trial, where they compared all of these commercial assays, about 5 different assays ranging from Oncotype DX to MammaPrint to the PAM50 assay to IHC4 and IHC4 Aqua. They compared the results of all those tests in a panel of about 313 newly diagnosed breast cancer patients. And you would expect that since all of those diagnostic tests have some ability to cull out low-risk populations from high-risk populations, you would think that they would all identify relatively the same types of patients no matter which assay you used. In fact, there was a paper published in the New England Journal of Medicine by Fan et al. Back in 2006 suggesting that probably most of these tests are able to cull out a low-risk group that probably would not benefit from chemotherapy, and therefore maybe it wouldn't matter which test you order. Maybe they're identifying the same patients. Well, in this OPTIMA trial, the opposite was observed when they measured all 5 assays and all 300-odd tumors. They found marked discordance from one assay to the next. For instance, the Oncotype DX score had the highest percentage of low-risk patients compared to any of the other assays. And so that calls into question the potential of each of these assays to truly identify all of the patients that are low risk. And based on that data, it was impossible for the authors of the paper to conclude which test was best. We don't know whether the Oncotype score perhaps overcalls low risk or whether the others are undercalling low risk, for example. As it happens, a number of these tests have been looked at retrospectively also for their ability to predict response to chemotherapy, not just prognosis, that the assays were built upon. And that introduces another problem, and that is there are a number of expression arrays that have been published in the literature now looking at specific gene expression profiles that predict for specific types of chemotherapy response, and yet none of those are integrated into our current armamentarium. All of the commercial assays were developed on prognostic signatures, not response signatures. So I offered a solution, perhaps one solution to this conundrum and the discordance in the Optima trial would be to take a step back and re-look at all of the bioinformatics and information and platforms that were used to develop the current generation of assays. And this was done by the Metabrick investigators and published just a couple of years ago. They looked at a set of 2,000 primary breast cancers, divided it into cohorts of 1,000, a test set and a validation cohort. And they used a new tool which actually integrates both the messenger RNA data and the copy number variation data. It turns out that in human breast cancer, the copy number variation data harbors a lot of the information on drivers of oncogenesis, particularly in breast and ovarian cancers, whereas in other tumor types, it's more collections of point mutations, single point mutations, that offer that same capability. And consequently, by integrating the expression array data with the copy number variation data, you get a more high-resolution picture of the phenotypes of breast cancer. And these are called integrated clusters. And there are 10 subtypes that fall out of that type of an algorithm, and they correlate with prognosis. They correlate with response to neoadjuvant chemotherapy. There's much greater granularity in their ability to predict chemo response as compared to any of the other assays that are currently commercially available. And so I think in future generations, it's maybe very likely that we'll look at these more highly integrated data sets that contain more of the molecular information, not just messenger RNA information, although the integrated cluster now is able to take a step back and show that some of their copy number combined data have gene expression profiles that correlate with them quite well, which sets the stage for perhaps future commercialization of that type of technology. It would be cheaper and easier if they just use the array data rather than the integrated data, and it looks like they have the capability to do that. So that's quite exciting. I think it's a look forward into the future development of breast cancer diagnostics. I think there's potential for more complete understanding of molecular phenotypes of breast cancer, and consequently that will inform better resolution in both prognosis but also treatment outcomes in our patients.